Hi, this is Miss Litton, and this is 7th period for Honors Bio. Say hi.
Hi.
Okay, so this is our chapter on development and aging. Big ticket item is how do we get from here, to forming a new organism. So, and then also we talked about aging. So I'm going to skip right to the key points as we go through here, and then we'll do all the questions as well. So the first thing I want to talk to you about is when fertilization takes place. And I want to remind you that the sperm wears a cap, and that cap is called a what? Acrosome. And what is inside that acrosome? I think you're saying it. What? Enzymes. Perfect. And those enzymes help digest something. What do they help digest? Zona pellucida. Perfect. And that allows the sperm nucleus to fuse with the egg nucleus and form a diploid zygote. Now, from that point, you have had a haploid and a haploid makes a diploid. From here on, now we need to take this one cell and make trillions of cells. So you can see, though there are modifications on this theme, in the simplest type of organism here, we can see the different stages. And so for your quiz, you're going to want to recognize the sequence of the stages, right? The sequence of the stages and what it looks like during that stage, you know, and the characteristics of that stage and any structures related to it. Okay, and we went over this pretty well in class, and we said, well, right after the sperm fertilizes the egg, we said cleavage, 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 which is just doing a bunch of what? Mitosis. But they don't take time at this point to do what? No, they have to replicate the DNA in order to do mitosis. Let's talk about the cell cycle. The cell cycle consists of interphase and M, right? Yeah. Okay, so here's our cell cycle, and the bulk of the time, the cell right here is an interphase, yes? An interphase gets broken down into G1, S, and G2, right? And then this is mitosis and cytokinesis, right? Prophase, metaphase, anaphase, telophase. So G1, right? G1, okay? Right here, that's when the cell is doing what? Growing. So they're going through the cell cycle, it's just they're blowing right through G1. Stop sign, no, done, okay? Blowing right through G1, and as long as the DNA is okay and it did mitosis okay, it'll just move right through that. So the cells aren't getting any bigger. So if it's taking up this much space right here to be a diploid zygote, 2 cells are taking up the same amount of space, and 4 cells are taking up the same amount of space because they're not giving a bunch of time for more cytoplasm, more organelles, and all of that, okay? So that's why when you look here at the size of this zygote, it's this big, it's still this big even though it has 2 cells, it's still this big even though it has 8 cells, it's still this big even though it might have 32 or 64 cells, because those cells haven't gotten bigger yet. You all right with that? Okay, so that is cleavage, mitosis, 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 right? And then it becomes a solid ball of cells, about the same size as the zygote was because there was no time for growth. That solid ball of cells is called a what? Solid ball of cells called a morula. So we're a morula first. So we're going cleavage, then we're going morula, solid ball of cells. Then we pump in what? Salt, and who follows? Water, and this way if water goes into it, we are a blastula. And what is a blastula? A blastula is a hollow ball of cells. What do you call the cavity, bless you, inside the blastula? Yeah, so the blastula has a blastocele, okay, inside of it. All right, now, then you have this hollow ball of cells, which we talked about like a red rubber playground ball, and it starts to enfold on itself like you could by letting out the gas. So we remember gastrula, okay? And so a gastrula is what comes next, and a gastrula is a layered ball of cells. So you're going to want to remember it's a layered ball of cells. And this is where you have that archenteron. And the opening to that archenteron, that's the blastopore. Okay, and then when you have a layered ball of cells, we just need to know the layers. Okay, gastrally have a layered ball of cells. The outermost layer is the ectoderm, the innermost layer is the endoderm, and the middle layer is the mesoderm. Okay, so that's what we have. And then the next stage after the gastrulus stage is what? The neurula. See them begin. See them begin. It's a new baby. See them begin. Okay, cleavage, morula, blastula, gastrula, neurula. See them begin. Got it? All right. So the neurula is when the notochord induces the spinal cord to form right above it. And I'll show you some pictures of that. Now, a difference, this is really on a landslip, very, very simple. In humans, though, inside your blastocyst, you'll have a small group of cells which will go through gastrulation, neurulation, and that's called the inner cell mass. So that's a variation on the theme. We also talked about how the amount of yolk you have influences how you go through specifically gastrulation, because if you have a whole lot of yolk, then you can't cleave that, and so you just leave it alone. Or if you have an intermediate amount of yolk, like a frog, you kind of grow around it. So I will show you that. But the gastrula is when you form those layers. Okay, questions on this? We know cleavage, morula is solid, blastula is hollow, the cavity is called the blastocele, gastrula is a layered ball, the new cavity is called the archenteron, and the opening to the archenteron is called the blastopore. What does the blastopore become in you and I? It becomes our anus, where in other organisms it might become their mouth. Good. Okay, and then the neurula. Clarifying questions. Ask now. Yes?
Can you explain the neurula again?
Yes, I'm going to hit that because it's not on this picture, but you can see it a little bit here, just so you know, in this section right here. Okay, so you're going to have, once the mesoderm forms, you're going to have a notochord, and it'll get the tissues right above it to fold in like this and form the spinal cord. And then the notochord will be the vertebrae that protects it. Okay, and I'll show you that on another picture. All right, so let me show you that other picture. Here we go. So here you're seeing the end, when you're all done, the 3 layers, you can see in blue and pink and yellow. But that process where you form the neurula, right here, okay, we can see the notochord in this beginnings of the mesoderm. See how the mesoderm isn't all the way around here? So here's our mesoderm. The notochord is here. The tissue above it is going to start to fold in on itself. And when it does this, we're at the organ level of development because we're forming our spinal cord. So that is that final stage there. Now, when we talk about layered, okay then, Papa. Here we go. When we talk about those layers, we know what each of those layers is going to be. So and it's easiest just to know what the outermost one is and the innermost one, and then any other system falls into the middle one. So the outermost layer, what did we say you get from the outermost layer? Skin and nervous system and the lens of the eye. Perfect. Okay? And the innermost one is going to be our tube inside of our body. What two systems are associated with the tube when you go in your mouth? Digestive and respiratory. So any associated glands, so if I say liver, you would say, oh, that's part of the digestive system. So that, not that I would just say liver, but we need to know systems. Digestive and respiratory systems are going to come from the endoderm. If I say musculoskeletal system, you're going to tell me, oh, that comes from the mesoderm. Reproductive, oh, that comes from the mesoderm, because it doesn't fall on our 2 bookends. Okay, so you want to be comfortable with that. So if I ask you about one of these layers, you'll know what you get from that layer. Your book also has a chart on it, but that's really, really detailed. If you just remember what we went over in class, epidermis, nervous system, lens of eye, innermost layer, linings of digestive, respiratory system, and then anything else that's mentioned is going to be mesoderm. Questions, concerns? Okay, let's do some questions. Josh, tell me. Sorry, I was checking my email. Oh, dang it. Okay. Okay, this person was really messed up. So let's go through it so we can help them out. Good job. What day is it? Boo, oh, that's mean. Okay. Now, I'm not going to say that one. All right. So see them begin. Could we do it that way, right? See cleavage first, and then morula, right? Next, and then blastula, and then gastrula, and then neurula. Okay, so tell me, just as a reminder, what is cleavage all about? What? Mitosis. Good answer. Because that was a wide open question. Dang it. Mitosis. Good. What is morula all about? Solid ball of cells. Good. Solid ball. Okay, what is blastula all about? Hollow ball. Hollow ball. And what's the cavity inside called? Blastocele. Good. Okay, and what's gastrula all about? Layered ball. And what are those layers? What are those layers? Ectoderm, what? Mesoderm and endoderm. I don't know what I was writing right there. Tell me what the ectoderm is going to be. Skin, nerve, lens. Okay, endo.
Musculatory skeleton.
No.
Respiratory.
Respiratory and digestive. And then this is everything else. Yes? Okay. And then what's the neurula?
Formation of spine.
Yes. Formation of nerve of spinal cord. Okay, good. Got it? All right, let's do another one. Did you guys see, just as a heads up, I put, in case you didn't get 100% on your, I put it in Google Classroom. Did you see that?
Yeah.
You have a chance to get 100%? You don't have to, but if you want to. Okay, so our Kentron, let's go back. Let's have a discussion. When do we have in our Kentron? Folding is into the gastrula when we have our layered ball of cells. So the innermost, where this has the blastocele, okay, the gastrula has the archenteron right here. The archenteron is going to be your gut tube. It's starting right here. It's either going to be your digestive system or your respiratory system. But the tube that goes all the way through, because my respiratory system stops right here, right? Mm-hmm. So the tube that goes all the way through, it's going to be part of my gut tube. So add that into your notes if you missed that. Okay, that's this one right here. Yeah, because what did some people say? They said vertebral column. What becomes a vertebral column? Notochord. Okay, and then some of you said none of the above. Okay, just wrong. Good job. Oh gosh, I can't say these out loud because this Mean Girls list is hideous. Let's get 100. Did we already talk about this? Mm-hmm. We did. Yay! 1. Oh, it's up there. Super proud. Again, Africa person. It's a terrible list. Shaka Khan. Okay, so the right answer is B. The blastula doesn't have the gut. The gut is coming from the archenteron. When do we have an archenteron? At the gastrula stage. So that's why it doesn't work. So you could say gastrula, and then it would work. Okay, and then somebody said C, gastrula, 3 germ layers. Yes, gastrula. What do we define that as? A ball of cells. What is it? Layer ball of cells. And then D, neurula, nervous system. Yes, the neurula is when you form your spinal cord. You're forming your nervous system. Check the person next to you. See if they got that or did they struggle a little bit. Yes, ma'am.
So the neurula forms the nervous system and the notochord forms the spinal cord?
When I talk to you about the notochord forming the spinal cord, that is an example of induction. When one tissue influences another tissue's development, it's just that the neurula, once you have a neurula, you have a spinal cord. So now you're in the nervous system. It's going that way. It's not like the notochord gets that pathway to happen. It's like this triggers this, triggers this. Just like we talked about, the gray crescent in the frog triggers gastrulation. The notochord triggers invagination to form the spinal cord. And when you're doing that, you're in the neurula stage. Just like when you have a layered ball of cells, you're in the gastrula stage. Does that make sense?
Yeah.
Okay.
Do you have that song on YouTube?
I don't. Do you want to film it?
Yes. Yeah.
Okay, are you going to do it with me?
Like right now?
Well, we can. Do you want to do it right now?
I guess so.
Okay. Well, then we can sing it right now, and then it'll be in here too.
Okay.
Okay, then.
I'm ready when
You got to do it on my phone because my phone's linked to my YouTube account. Pictures, pictures, pictures.
I'm gonna be so good at it. Where do I film?
Just be right there. Okay. Let's see if I remember it. My hair look funny? Okay. Oh, ready? Okay. So we're talking about development. So here comes Mr. Happy Sperm, and he's wearing a cap. What's his cap called? Good. And what's inside the acrosome? Enzyme to help us digest the zona pellucida. Wait, what do you say?
Shouldn't I? Oh wait. It says shut off. It shut off.
Oh, my battery must be
Do you
dying.
want me to do it and send it to you?
Yeah. I wonder if I can get it. Well, can you send it? Or should we go plug in my phone?
I can send it. I can just text it to you.
Okay, and it'll come?
Yeah.
Okay.
Okay, wait. Ready and go.
Hi, we're talking about development. So we're going to start right here. Here comes Mr. Happy Sperm. And what is he wearing on his head? Acrosome, a cap. And what does it contain? Enzymes. And the enzymes help digest the zona pellucida of the egg so that the sperm nucleus and the egg nucleus come together and you form a diploid zygote. The diploid zygote does cleavage, 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 which is a whole lot of what? Mitosis, and we form a solid ball of cells called a morula. And then we pump in salt. Who follows? Water. And now we're going to form a blastula, which is a hollow ball of cells. And the cavity inside is the blastocele. Then we do invagination. We fold in, like letting out the gas, and we're forming a gastrula, which is the layered ball of cells. The outermost layer is called the ectoderm. Innermost layer is the endoderm. Middle layer is the mesoderm. The ectoderm will form the what? Skin and your nervous system and the lens of the eye. The endoderm will form what two systems? Digestive and respiratory. And the space inside, when we form our gastrula, is called the what? Our archenteron, and that will be the gut tube, right? And the opening to it is called the blastopore, which will be our anus. Good. After the gastrula, you're going to form the neurula. And the neurula is when you form the beginnings of your nervous system. The notochord induces the ectoderm above it to fold in and form the spinal cord, and then the notochord will be the vertebrae around it.
Yeah, that was good.
Yay!
Okay, after I can.
Yes, no problem.
So during the morula stage, when you do cell division, how do you do cell division without growth?
They just, because to do cell division, all you have to do is replicate the DNA and have the enzymes to facilitate it. So you don't actually have to get bigger.
Okay.
Yeah. Now, they already left, but I want to tell you, you need to know these stages that I'm going through. You also need to know those stages of development that we discussed and the stages of birth and the theories on aging.
Wait,
Okay.
is
Yes.
that from chapter 23? Or is that still from this chapter?
From this chapter.
Okay.
Yeah, it's all this chapter. I have a tech-like meeting today? What? I did not know that. Oh, okay. Okay, so D, the lens comes from the what? Ectoderm, which we just reviewed. The heart does come from the mesoderm because it doesn't fall underneath the skin, nervous system, lens of the eye, ectoderm, and it doesn't fall underneath the digestive or respiratory system that you would get from the endoderm. So heart does come from the mesoderm, and the gut does come from the endoderm. Remember, the gut tube is coming from that innermost layer. All right, differentiation. We definitely need to make sure we discuss that. In differentiation, we talked about how is it that you have so many specialized cells coming from just one zygote. And what we said is the DNA is the same in all of these cells, but what's in the cytoplasm can vary, the concentrations of the proteins. And when we looked at this very simple picture, we said we can see all the red and green, quote unquote, proteins, but after the very first division, after the very first mitosis, we have more red where? In the top or the bottom cell? Bottom cell. So what's in the cytoplasm can influence which genes are expressed. And what we later learn in this discussion is you call the proteins morphogens, and the DNA that is really critical are the homeotic genes. So because you have different concentrations of them, those variations, and it can lead to different differentiation in those cells. And we looked at the frog, but if you remember, we did a simple picture like this. And we said, okay, the sperm fertilizes, where the sperm fertilizes the egg, what end is that going to be? The head. What's another name for that? Anterior. So if this is the anterior region, then this by default is going to be the posterior. Okay, so, and then the other one we did is we talked about that this area here that has more yolk, where you have all of this yolk right here, this is going to be what region? Do you remember? This is the ventral region, the front, and that's called the vegetal pole. Not vegetable, but vegetal pole. It's going to be the front of you. And if this is going to be the front, then this is going to be the what? Dorsal. And this is called the animal pole. So right away, when you're doing these divisions, it's like you could see this one has sperm, this one has sperm and yolk, this one has just yolk, and this one has nothing. So we already have differences that set it up once cytokinesis takes place, once you go through mitosis. Differences is what in the cytoplasm, what's in the cytoplasm is going to influence what DNA is expressed. And we looked at different experiments with frogs, etc. Now, morphogenesis is playing out that expression. It's the movement of cells that changes the shape and form of the body parts. Morphogenesis is when you start to form those body parts. What do you call the things that get it to happen. They're called morphogens, right? Morphogens make morphogenesis happen. That's the chemical. If one cell secretes a morphogen that influences the development of another one, that's called induction. Yeah. So when tissues differentiate, okay, so we have tissue differentiation. When that's taking place, that occurs by a morphogen, right, is causing that. The process where one tissue influences another tissue, that's called induction. So the thing releasing, so here we could have tissue number 1 releasing a morphogen affecting tissue number 2's differentiation. This process right here, where one tissue influences another tissue, that's induction. What do they induce with? They induce with a morphogen. What does the morphogen actually impact? It impacts certain regions in their DNA, and those are called homeotic genes. That's what that morphogen actually influences, are these homeotic genes, and then that causes them to differentiate down a specific pathway. The process of one tissue influencing another is induction. What do they use to induce them? A morphogen. Those morphogens affect this tissue number 2's genes, his homeotic genes, which cause him to differentiate. Now, tissue number 2, now they can secrete another morphogen, which affects tissue number what? 3, and it might actually go back and influence back on tissue number 1, okay, on their homeotic genes in their DNA. Okay, and that's how this can trigger this, can trigger this, can trigger this. Big, got it? Okay. And that's how you see segmentation in organisms. Okay, and then this just was saying, here's a morphogen influencing a homeotic gene, which then makes another morphogen, which then influences another homeotic gene. We looked at an example of induction, and that's when the notochord influences the epidermis above it to fold in and form the spinal cord. Okay, then we said apoptosis is a normal part of development. Death is a normal part of development, and that helps carve our organs out, like our fingers and our digits and our bones, by the death of other cells. Just like if you were going to make a beautiful clay pot, you would need to carve off some of the clay and take out some of the interior part. So that's a normal part of development. Okay, because we're running out of time, let's just buzz through these. Genes that control the structure pattern of an animal are called what? Homeotic genes. Okay, those are the genes, right? Number 2, the ability of one embryonic tissue to influence another tissue. What is that called? Induction. I'm influencing you. Okay? This word morphogenesis means what comes as a result of your influence. My organs developing. Pattern formation might come as a result of that influence. Apoptosis might come as a result, cellular death. Cellular differentiation is as I specialize. But the process of this tissue influencing another, like tissue peer pressure, that is induction. Morphogenesis is associated with protein gradients, those morphogens in a gradient. It does have to do with induction. It does shape and form the body, and you are going to use homeotic genes to code for those morphogens. So morphogenesis, and you have to talk about all those things to talk about morphogenesis. Programmed cell death, is it called induction? No. Is it a necessary part of development?
Yes.
Yes. Is it called reduction? No. Is it normal for these things? Yes. Is it called apoptosis? Yes. Only occurs when people age? No. Before you were ever born. If it didn't happen then, and you wouldn't, have your digits, your fingers, your radius, your ulna. Okay. So the next part you want to be familiar with, okay, is I'm going to just jump to the summary slide since we're running out of time, is what happens in human development, right? Within week 1, remember we say you blast into the first week, and then you can blast on into the uterine lining. You begin implantation, you don't finish it. Week 2, remember how we remember number 2 might be associated with some gastrulation. Not to be gross, but you'll probably remember it. Week 3, we took the 3 and we turned it sideways to remember N, cursive N, neurula, and also a what? Heart. Okay, so week 3, neurula and the heart begins pumping. Weeks 4 and 5, you have 4 limbs, and we think about the 5 senses. That's how we remember weeks 4 and 5. That's when it's developing. Okay, months 3 and 4, you're getting bones, male and female. And then week 10 is the placenta. Bones and male and female. How are you going to remember that? Month 3, month 4. Bones, month 3. And we're in the months before you get bones. Before that, what is it? Cartilage. Yeah, it would be cartilage before it becomes a bone. And then months 3 and 4, male and female. Okay, and then months 5 through 7, by the end of that, you're going to survive. Eyes open, fetus can live. So you just want to, these are like straight up memorization ones. So just don't confuse your weeks and your months. Okay? All right, and then looking at the questions, implantation begins at what stage? Yeah, blast into the first week and blast right on into that uterus. Similarities in the development of birds, mammals, and reptiles. Remember we looked in class, we looked at the different membranes. And remember, what's the membrane that goes right around the baby? You've heard of it. They talk about it all the time. Amniotic. Like the amnion, when somebody's water break, it's the amnion that's right around the embryo, okay, is the amnion. The outermost membrane is called the chorion. A is closest to the baby. Okay, and then here. When does the neurula form? Week 3. How are you remembering that? Take that 3, right, and turn it sideways, right? And then you remember that, and then make a heart out of it. Okay. Good. When can you tell male from female? 3 to 4. They're knocking on the door. I don't know. I'm trying to think of ways for you to remember that. When does the morula form? Week 1, because you blast into the first week. So what are you before you're before the blastula? You're the morula. So week 1. Good. When do the limb buds and senses form? And you're remembering that because 5 senses. You have more of the senses than that. Birth. You want to know your stages of birth. Okay. First stage is that cervix has to dilate. Second and third stage are both outs. Okay. And actually something's going to happen, come out all 3 times. The first stage, the cervix dilates, water comes out. Yeah, water breaks. Second stage, a baby comes out. And the third stage, a placenta comes out. So something comes out every time. Water, baby, placenta. Okay, if you think about it like that, that would probably make sense to you. Okay, last but not least are your theories on aging. So one stage is genetic origin. You come from a line of long-lived people, you're probably going to live long. The other one is your whole body just kind of starts shutting down. Third one is extrinsic factors have the biggest impact. And probably that would be what most people would agree to this, is that how you treat your body, how you live your life is going to determine how long and healthy of a life you have. Which theory of aging states that hormones start to decline and it affects other systems? Other systems. Whole body process. Good. when attempting to age well, you don't need to worry about what you do when you are younger. True or false? False. You don't have to worry because scientists will find cures for the diseases you might give yourself. False. We'll want to have a lifetime of good health habits like making time for regular exercise and eating good food. True. We'll want to do many different drugs as possible and then become addicted to most of them. False. So glad you got that one. Eaten. Thank you. Yay. Have some toast.
Do I really have a